Many bacteria, such as E. coli, have dimensions of about a micrometer. At the cell's centre is a bundle of DNA known as the nucleoid. Proteins that perform essential functions, such as copying and repairing this DNA, move around the nucleoid searching for suitable locations to bind. Through fluorescence microscopy, we can image both the nucleoid and these essential proteins as they perform their functions. Fluorescence microscopy directly images proteins in cells utilising fluorescent probes, which absorb one wavelength of light and re-emit another. Genetic modification of the cell's genome enables these fluorescent probes to be attached directly to the proteins of interest. This technique can be applied to most of the 4,000 different proteins in bacteria. For example, E. coli cells contain tens of thousands of copies of a protein known as HU, which binds to all parts of the nucleoid. By imaging this protein, we indirectly observe the nucleoid shape and can visualize how antibiotics, like rifampicin, a major drug used in the treatment of tuberculosis, decompact the bacterial nucleoid. To observe processes occurring at the single molecule level, we use an imaging technique known as PALM. PALM uses probes that are initially non-fluorescent until a second light source causes an irreversible change to the molecule's structure, causing it to become excitable. Excitation of the photoactivated probe causes it to re-emit light until it gets destroyed. By controlling the intensity of the photoactivation light source, we can ensure that there is typically only one molecule emitting light at any given time. The light emitted by a single immobile molecule appears as a Gaussian-shaped spot on our image known as the PSF, or point spread function, whose shape is defined by the microscope optics. By fitting the known PSF shape to the spot, we obtain the molecule's position with an accuracy of about 40 nanometers. Connecting localizations in successive frames, we track molecules and study their diffusion properties. For example, we track RNAP, a protein involved in copying DNA information into a messenger molecule to synthesize proteins. The RNAP molecules are observed to have two diffusive states: a fast-moving state, in which the protein diffuses around the nucleoid, searching for a DNA site to bind, and a second state, in which RNAP is bound to the DNA whilst transcribing genes. These techniques provide greater understanding of fundamental mechanisms in biology, such as the mode of action of antibiotics, and enables us to develop better synthetic biology and biomedical tools.